I'm delighted that Marta Soares could join us today. So I was going to say she's joining us from the University of York, but I believe she's joining us from her bedroom. At York, she's a senior research fellow in the Centre for Health Informatics. She's currently a member of the National Institute for Health and Care Excellence Technology Appraisal Committee. She's got really interesting background across some industry and academic experience, so I think that always makes for an interesting talk. And it's also worth mentioning that in 2014 she received the ISPOL Award for Excellence in Application of Pharmacoeconomics and Health Outcomes Research. So I know Marta's going to tell us a little bit about her background as well as part of the talk. So at this point, I'll hand over to her.
Well, thank you. First of all, thank you for the invitation to present. I hope everyone can hear me well. So I'm going to give you a bit of a break from COVID research, although to be honest, this is related in a way because lots of the policy decisions have been based on judgments. So I'm trying to share my screen now. I hope everyone can see my screen. Perfect. So I was saying lots of policy decisions have been highlighted to be based on expert judgment, obviously under the context of uncertainty. So I'm hoping that this research, which was conducted before COVID, is still relevant for COVID and a number of other setups and contexts. So I'm going to talk to you about a couple of experiments, or 3 experiments that were developed to study how accurate individuals are in judging uncertain quantities. So this has basically been termed as expert elicitation under uncertainty. And we use it in a very specific policy context, but the research I'm presenting here is very generic anyway. But I just wanted to give you a bit of context of my context, a bit in lieu of an introduction. And the context that I work on is health economics. We obviously, you'll see ahead that we use lots of data and you're data scientists, so you'll relate to that. But obviously, you'll also relate to the fact that sometimes there is no data available to answer some relevant questions. So this is the kind of overall context of this research. In terms of health economics, as the name kind of highlights, is a discipline of economics applied to health and social care. So obviously motivated by growing demand, this discipline for growing demand for quality and cost-effective health and social care, this imposes pressures on healthcare systems, and this is kind of what motivated the start of this combined discipline, maybe around 50 years ago. So we typically inform policy decisions in health. So one of the aspects, I don't know if you can see my mouse pointer. I hope so.
Yeah, we can. Yeah,
perfect.
That's fine.
Yeah, so I'll need it for later on anyway. So health economics informs policy decisions such as choice of treatments, and they're not necessarily always treatments. It could be diagnostic tests, etc. Any resources that the NHS uses. And this is termed health technology assessment. The assessments required to inform this choice of treatment strategies, healthcare resources. But it also informs the way health and social care systems should be organized, paid for and monitored, or how to reward and safeguard the workforce and community volunteers that are involved in health systems. I've highlighted this first option because this is mainly my kind of area of work. And I'm going to give you a bit more of an introduction to this. So in terms of health technology assessment, so this basically involves resource decisions, as I've mentioned before. So that's not just which technologies to use or strategies, but also in whom, when. So when meaning in which point in the disease pathway, and where, typically primary care, secondary care decisions. So they're very complex kind of decisions about treatments and strategies on the use of resources by the NHS. So what we typically use to inform these assessments, these health technology assessments, is what we call cost effectiveness analysis. in which we aim to compare alternative strategies in terms of the health effects and the cost implications. So specifically, we look at long-term effects on population health. So these typically include life expectancy, but also weighted by the quality of life, the morbidity that lifetime is lived in, and also total cost implications for any stakeholders or individuals. So we kind of look at these two dimensions, so the health effects and the costs, and the economics kind of discipline provides a way of making decisions based on these two often competing dimensions. So often you have an intervention that increases population health, but also increases costs for the NHS. So how does the economics make sense of this balance? So it does so by considering, just forget about the health effects, so just considering the cost implications. And what economics does or provides is a framework to consider the health impacts of these costs, these additional costs that might be imposed on health systems. So these additional costs will have implications for these or other patients in the health systems that see those resources not used for other purposes. So this has been termed health opportunity costs, and you can discount these health losses from the health opportunity costs from the total health that a particular technology might bring. So this is the contribution of economics. So this is just a bit of context. So obviously for these types of decisions, and you might have seen loads in the news about cancer patients being deprived from a particular treatment because it hasn't been approved for use in the NHS, etc. So these are the dimensions that typically underlie those types of decisions. But for that, for that you need evidence. You need not just data, but other types of evidence. So evidence lies at the heart of these types of decisions, which are typically very explicit, at least in the UK, they are very explicit. So there's an institute, the National Institute for Health and Care Excellence, that facilitates a decision-making process, which is collective. So there's a group of people that are involved in those decisions, and they base those decisions on evidence, on existing evidence. So obviously, to ascertain long-term effects on population health and total cost implications of alternative treatments, you need evidence, and you need typically multiple sources, because you need evidence on different aspects of disease and treatment, and you need to be able to consider this evidence together and extrapolate it over time. So there's a need for modeling, typically. So in this context, what we have is a model, any model, which imposes a parameter space plus some additional structural uncertainties, which you probably are familiar with. We will have an evidence space, and we need to match, right? We need to use this evidence to inform our parameter space. So the evidence space will have some empirical evidence, some data on a number of studies or databases, and we need to use that evidence, analyze it statistically to parameterize this model we need to develop to determine the long-term costs and qualities of alternative interventions. We also use mechanistic evidence to inform judgments about model structure, about the duration of treatment effects, for example, or other assumptions underlying the model. And we typically might also need judgments to describe uncertainties over parameters for which there is no evidence, no empirical evidence on. So that's one of the aspects of expert elicitation that is relevant. Obviously, given the explicit decision-making context, the breadth and the quality of the evidence available is also important, and that's kind of beyond sampling uncertainty. So the evidence might be less representative of the context of interest, for example, of the UK, if we're making a decision for the UK. It might be sparse, it might have low, medium or low quality, so not be high quality evidence. There might be other sets of evidence that are also relevant, but they're not direct on the problem we want. So for example, there might be evidence on adult patients when we're trying to make a decision on pediatric patients. That might be relevant, but not directly relevant. And the evidence might also be inconsistent, given that we might be looking at multiple sources. These might present inconsistent results that we can't directly interpret based on the characteristics of the data sets. So you can see that, so these later sorts of issues with the evidence, with the data, are difficult to quantify as uncertainties. So we might also use expert judgment here to try and quantify these additional uncertainties that we can't represent easily from the data we have. We can use expert judgments, as I mentioned, to reflect uncertainties for which there is no evidence, or we can use expert judgments to characterize some of the structural uncertainties in our kind of parameter space, the design of our model. So one of the things I focused a lot in this last slide is about uncertainty. And obviously uncertainty is pervasive, as you know. In terms of the decisions we make today, the decisions about particular interventions are typically based on expected values, on means. And this is because, so this is a bit different from the kind of inference context that statisticians typically consider. So we typically decide based on expected values, and this is because rejecting an intervention that is expected to be beneficial solely on the grounds of uncertainty is really not ethically justified in the medical field and in many decision fields, to be honest, so it's not just in health. However, we need to think that under uncertainty, further research that's timely might be considered, and this might be useful to reverse any initial guidance if this initial guidance is proven wrong by the research, and in this way kind of avoid the consequences of uncertainty to patients and health systems. So I've talked a bit how expert elicitation might fit in this decision context, but from this point onwards, everything will be general, to be honest, not specific to the context of health economics or health technology assessment. I just wanted to give you a bit of background. So critically, the main thing is that expert judgment is always required for decision making. And so we need to make sure that when we collect these judgments, when we collect this evidence, I'm not going to call it data, I'm going to call it evidence. When we collect this evidence, we minimize biases that might be typical of these. So in the context for today, I'm going to define expert elicitation as a systematic process of formalizing and quantifying, typically in probabilistic terms, expert judgments about uncertain quantities. So we're typically interested in uncertain quantities, so we'd like to elicit distributions, which I'm assuming everyone will be familiar with. So these are obviously core concepts to Bayesian inference. For those of you familiar with Bayesian inference, where subjective priors are typically used to update any existing data. They are formally used in support of decision making in a few a few areas. So for example, the European Food Safety Agency, EFSA, that I mentioned here, has released a few years back, 3 or 4 years back, a document with formal support for expert elicitation, for structured methods of expert elicitation. One of the important things here is that uncertainty has a slight different definition to what we're used to. So uncertainty here reflects the degree of belief over the uncertain quantity. So it reflects imperfect knowledge, also called epistemic uncertainty in psychological research. But we're here trying to reflect imperfect knowledge. And as I've mentioned before, some good elicitation. The objective is to minimize biases. So these can be motivational biases, so the kind of conflicts of interest type biases, but they might also be more relevant in a way, because we can't control them as well, is cognitive biases. So cognitive biases are kind of shortcuts we all use to make judgments, which heuristics are cognitive shortcuts we use, and these might result in biases, so in systematic biases in the judgments we give. Obviously, we need to acknowledge that all probabilities elicited should, if they reflect personal knowledge, which will be imperfect, they are personal, so they are individual. So I've mentioned a few of these items in this slide. So possible uses for elicitation. I've also mentioned this in the previous slide. I have a few references here that are not fully referenced. So if anyone would like to know more about these, just drop me an email and I'll forward you the full reference. But basically, as I mentioned before, elicitation can be used to generate initial estimates when empirical data is absent, but it can also be used to generate weights for alternative structural assumptions or bias correction if we have evidence that might, we suspect it's biased. It can help characterize between study heterogeneity when you have multiple sources of evidence and you can't identify heterogeneity clearly. And it's been used often for sample size calculations for studies. So the references here are for the health technology assessment application methods developed for health technology assessment, but there are applications of elicitation in other areas which cover similar kind of parameters and aspects of data and evidence. So critically, the motivation for this work I'm presenting today. So critically, when you ask someone to elicit a particular quantity, this requires a number of decisions about aspects of the design of this data collection, this evidence collection, correct evidence collection process, design, conduct, and analysis. So there are a number of methodological options in the literature, a number of choices. And there are a number of also a number of protocols, so kind of guides to good practice that focus in more than one of these choices. Some of them are generic, so some of them are driven by a generic, for example, statistics literature, like the example here, the Sheffield Elicitation Framework or SHELF. And some others are domain specific, like I've mentioned the European Food Safety Agency protocol. The problem is that there's little research to support any of the design, conduct, and analysis choices that need to be made. And there's obvious reasons for this. So one of the main reasons is that accuracy can't really be measured, as expert beliefs are inherently unobservable. So we can ask them what they believe about a particular quantity, but we can't really determine whether they're being accurate in representing their own beliefs because we can't observe them. So what happens is that the existing methodological research around elicitation is non-committal, typically in the conclusions it makes. There's lots of inconsistencies, there's different experimental setups, different designs of the research, so they might return inconsistent results without a clear kind of reason, and most of it is actually poor quality. So we've designed these experiments, which kind of are very neat, I think, in my opinion. They go back to basics, and you'll see how in the later slides. But basically this work has been motivated and conducted within an MRC funded project. So that's Medical Research Council. They have a method stream, and we have managed to secure funding for developing a protocol for expert elicitation, particularly in healthcare, what we've called healthcare decision making. And the experiments were conducted within this work. You can see there's lots of other people involved in this research as well. They're named here from different institutions. So the report has been written and is awaiting publication, but it's already available on White Rose. So I'm focusing today just on the experimental work, not the remaining of the project. And we have implemented basically 3 experiments. So the first one compares, and I'm going to go through these in detail in the next few slides. So this is just an overview. So the first compares 2 methods of eliciting uncertainty. The second experiment determines whether individuals are able to extrapolate from their knowledge base to different populations, and this will all become much clearer in the next few slides. The third experiment explores how individuals revise their probability assessments when presented with Delphi type group summaries. So I'll talk a bit about the Delphi ahead as well. So how have we set up these experiments? So we developed these for purpose. So they were based on a paper published by Wang in 2002, and this paper uses what they call a simulated learning process. So we've kind of adopted a similar approach. So what we've done was to use simulated observations, which were generated by us and recorded, to determine the knowledge of an individual. So this is obviously reflective of learning in kind of healthcare. So for example, doctors observing patients as they come into clinic. Other contexts might not use as much of this learning by observation, but we'll talk about it at the end. But basically, this is a learning by observation type setup. The data we feed participants determines their belief fully. So we've made that was one of our aims for the data to determine the individual's beliefs. So we could be able to measure accuracy. So the elicited distribution versus the posterior distribution of the data they've observed, plus some prior beliefs that might be vague, could be compared to each other to determine accuracy. So an experimental approach such as this, as I mentioned, might be a bit simplistic to what you'd expect in asking an expert about a particular quantity, but it does allow defining the conditions for the experiment and to isolate determinants of accuracy in this case. So we can test specific hypotheses and we can standardize the task to reduce between individual variation that would be expected in a more naturalistic setting. So this means that whilst we did impose randomization, we are expecting to require a smaller sample size than if it was a more naturalistic kind of setting for the experiments. So one of the critical or the important issues to recognize here is that because we've gave individuals the data, the information, we gave individuals information, we are testing, basically what we're testing here with these experiments is their ability to extract information from observation and quantify their beliefs in probabilistic terms. So this is, we've termed a sort of a probabilistic accuracy. So as I've mentioned, participants were shown random observations from a statistical model, and to try and not let external priors and information influence their elicited distributions, we framed this as an abstract, a very abstract medical problem. So what we've presented to the individuals was a hypothetical kind of scenario, and the participants in the experiment were asked to act as kind of practitioners for a number of clinic days. So they saw a set of patients with the symptomatic hypothetical disease. In each day, the participants would care for kind of a variable number of patients. Let's see if I can, yeah. So for example, this is how we've represented it. So in this particular day, in the simulation, there were 11 patients in clinic. So the number of patients in clinic varied between 6 and 13 and was randomly selected. And participants were presented with 2 pills of different effectiveness. They weren't told which pill was which, so they had tags like pill A and pill B. They wouldn't know which one was the less effective at the beginning of the game, but they knew that there was one pill that was less effective than the other. So they could choose the pill to use. And once they chose the pill they would use that day, a new screen appeared showing how many of the patients had cured their symptoms, were relieved of symptoms. So in this case here, represents that after treatment with pill J, the pill that the participant chose, 5 patients were relieved of symptoms, and there was this display that would appear in the screen. So after observing a number of clinic days, participants were asked to elicit the effectiveness of the most effective pill. So this was the target question for the elicitation. So the 2 pills, just to clarify, the 2 pills were implemented here for engagement. So the first thing was that this was a game. They would have to find out which pill was more effective so that they could elicit on that pill. And that allowed not only to increase engagement, but also to determine an incentive scheme. So we gave the participants monetary incentive, which was personalized, and it depended on how fast they would find out which pill was the most effective and the accuracy to the elicitation. So it would incentivize engagement to the game and accuracy in the elicitation. So the target question for experiments 1 and 3, not 2, but for experiments 1 and 3, the target question was exactly on the quantity they've observed. So it asked about pill X, the most effective of the 2 pills. If you were able to treat all patients in the population with this pill, what proportion would you expect to become symptom-free? So that was a target question. So we used 2 methods of eliciting uncertainty. I'm assuming you're not aware of these methods, but basically there are a number of methods. The most used methods are these 2. So one of them is the bisection method. The bisection method asks experts to elicit directly 3 quartiles of their distribution. So you can read this in your own time, or maybe I'll read it out loud. So the median is asked as, can you determine a value, your midpoint, such as the proportion is equally likely to be less than or greater than this value? The 25th percentile, or the first quartile, is worded as, suppose you were told that the proportion is below your assessed midpoint, can you now provide a new value, your lower quartile, so that the proportion of patients is equally likely to be less than or greater than this value? And finally, the third quartile was phrased in this way. Suppose you were told that the proportion is above your assessed midpoint, can you now provide a new value, your upper quartile, so that the proportion of patients is equally likely to be less than or greater than this value? So it's basically, as the name refers, it's by its sections the range of values into half, so the median, and then from the lower half, it sections again into half to ask for the first quartile, and then from the upper half, it sections to get to the third quartile. So the alternative is the chips and bins method. So the chips and bins method is partitions the interval of values we aim to elicit on into a number of bins. So typically 20, so a large number of bins, and then asks individuals, participants, to place crosses or chips on the values they believe to be most, the values they believe to be true. So basically they're drawing a histogram really. So the more chips they place, the stronger is their belief that the true value lies within that interval. So in terms of design of these experiments, so because we are building up the knowledge base of participants, it was more important for us that they were representative of the type of kind of skills we expect from experts in healthcare. So we've recruited students from the University of York with a clinical background or undergoing clinical training. So at York, we have medicine, midwifery, and nursing, all 3 degrees. So we've recruited from those areas. In terms of the conduct, to facilitate delivering training, because eliciting uncertainty isn't that straightforward, we had face-to-face meetings. Each lasted for 2 to 3 hours, so including 1 hour of training. As you might have observed from the displays I've shown, the experiments were delivered using the Shiny for R. We've extensively piloted these as necessary, and as I've mentioned, we gave the participants some financial incentives with as I've mentioned before, based on engagement to game and accuracy. So in terms of outcomes of our experiments, of metrics, we obviously wanted to compare the elicited distribution with a true, what I'm here calling the true distribution, so the posterior of the data with some vague priors. And we clearly, obviously in comparing distributions, there's always 2 dimensions, bias and uncertainty. So in terms of bias, reflecting the difference in the means of the elicited versus the true distribution. And in terms of uncertainty, we've used the ratio of standard deviations of the 2 distributions. So everything I'm presenting relates to the elicited versus true distribution. We've tried to find a metric that would combine both bias and uncertainty, and we've used the Kullback-Leibler divergence measure, which is a measure of information lost when one distribution is approximated by another. The results weren't that straightforward with the Kullback-Leibler divergence, so we can talk about it at the end if that's of interest, but I'm not presenting any results here of this metric. We've collected preference for alternative methods as well. So we aimed for 64 participants and we got 72. So you can see the kind of split of individuals here in the slides. They were obviously young, mean age 22, mostly female, and there were 20%, 15% of postgraduates, 19.4% of postgraduates. We've collected some measures based on psychology, so a numeracy score and a decision style inventory. We haven't analyzed the data on these yet, on how these affect the judgments yet. So we haven't analyzed this data. So the first experiment aims to compare 2 alternative methods of elicitation, as I've mentioned in the overview slide, and these were the bisection and the chips and bins that I've shown you. So in terms of the literature, it's generally thought that the bisection method, so the method that elicits the quartiles directly, returns wider distributions than the chips and bins. But the chips and bins method is thought of as being more intuitive for participants to complete, particularly the types of individuals we're looking for, which have maybe less training in quantitative skills, at least on their base training. As you've seen from the setup of this experiment 1, the target question was exactly what the individuals observed. So the proportion of patients with symptom relief on a particular pill. So we don't really expect bias here to be very different. We're looking mostly with this experiment at uncertainty. And to do so, we've defined 2 uncertainty scenarios. And these change so two uncertainty scenarios. One is high precision and one is low precision. That's how we've called them, but not necessarily an absolute criteria. It's a relative criteria, right? So in the terms of, on the high precision scenario, the participants observed more clinic days. So they observed 25 clinic days. And they had less variation in outcomes between days because we've basically used the binomial model to simulate the observations they saw. In the low precision scenario, they observed less clinic days, so they observed only 10 clinic days. And we've sampled from a beta binomial model, so there's some over dispersion here, so there's more variation in outcomes between days. So the probability is varying every day. So this is how we've defined our two scenarios. So the design of our experiment, so this was a repeat measures design. So each participant elicited every single one of the 4 combinations of the 2 levels of precision and the 2 methods of elicitation. They've elicited each of these 4 combinations with different probability values. And to implement this experiment, we've used a full factorial design using a 4x4 Greco-Latin square. So in terms of results, so as I've mentioned, each of the 72 participants elicited 4 quantities. So that's a total of 288 distributions elicited. So I hope you can distinguish the black and the gray points here. So just to go through the plot, the graph here. So we have bias. So these results only concern bias. And you have on the x-axis bias, so absolute, not absolute, natural scale bias. And in the x-axis, you have bias with the chips and bins method. And in the y-axis, bias with bisection. And this is under the same precision scenario. So you have 2 points for each person. So 1 point will be a high precision point and another point will be a low precision, a gray point. The blue dots represent the mean across the individuals on the low precision group. Sorry, in the low precision group distributions, the low precision distributions. And the red dot will reflect the mean on the high precision distributions. So you can see when bias is 0, obviously the participants elicited a mean for their distribution, which reflects exactly the mean of the data they've observed. And the same here for the 0 on the y-axis, which reflects the bisection method. So in interpreting this plot, you can see that the results show widely scattered points. So this means that there is large variability in the responses. However, the mean and the median as well, which is not shown here, is close to 0. Are both close to 0. And the points are symmetrically dispersed around the value of 0, which suggests participants are relatively unbiased. And as I've mentioned before, this was expected because we're eliciting directly on the target question, which they've observed. However, you can see that in the high precision scenario, in the low precision scenario, the gray dots, there's a bigger dispersion of values, which basically means that absolute bias is higher on average in this scenario. Bias isn't very interesting here. Again, no evidence of bias across the 2 methods. So across the 2 scenarios and across the 2 methods. There's no evidence of bias here. So the results on uncertainty are much more interesting. So the x-axis here, so the graph is quite similar in design, but the x-axis here represents the standard deviation ratio. So note that the labels are in the natural scale, despite the actual scale is in the natural logarithm scale. And we have, so the standard deviation ratio in x-axis for chips and bins and in y-axis for bisection. So a standard deviation ratio of 1 means that the individual elicited, the standard deviation of the elicited distribution is exactly the same as the standard deviation of the true distribution. here on chips and bins, and here on bisection. The diagonal line here just reflects an equal standard deviation ratio, SDR, for both bisection, chips and bins, and bisection. So points placed close to this line have the same SDR for bisection and chips and bins. Now, in interpreting this scale, so an SDR above 1 means that the elicited distribution has a higher standard deviation ratio than the true distribution. So that means that it's underconfident. And an SDR below 1 means that the standard deviation of the elicited distribution is smaller than the standard deviation of the true distribution, so it reflects overconfidence. So what we can see here from this plot is that there is, the first thing we notice is that there is a separation of the points on the two scenarios, so of high precision and low precision. High precision, the mean of the high precision elicitations has a standard deviation ratio above 1. So that means that the responses are generally overconfident. Sorry, I mean underconfident. And for something was not right on my statement. So in the low precision scenario, the SDRs are below 1, so that means that the elicited distributions are overconfident. So that basically means that individuals don't adjust their representations of uncertainty sufficiently on these two scenarios. So you can see that the blue dot, if we start by the blue dot here on the low precision scenario is quite close to the diagonal line. So that reflects that the methods are performed similarly on average. And the points are kind of distributed on both, proportionally on both sides of the diagonal line. Now in the high precision scenario, the mean is a bit above the line, which means that the bisection methods returns even more underconfident answers than the chips and bins, which is already underconfident in this scenario. And this is supported by a much higher proportion of points above the diagonal line here. So those are the kind of the results, main results of experiment 1. We've also asked individuals whether they prefer the chips and bins or the bisection. And most of them responded to prefer the chips and bins, although some of them prefer the bisection. So in the second experiment, we've tried to explore whether individuals that have a different knowledge base to the target question and hence need to adjust their beliefs in order to respond to the target question, how their accuracy, if they were accurate in doing such extrapolation. So just an example here, and this is what we're going to use in the experiments. Imagine you have patients with 2 levels of disease severity. So you have 2 subgroups of patients. One subgroup is more severe than the other. A particular doctor or healthcare practitioner can have a case mix such that they observe a higher proportion of more severe cases than you'd expect in the overall population. So if you ask this particular doctor, what is the prognosis of these patients in the overall population, they have to adjust their experience to the expected split in the general population. So obviously, for this example, the individual's knowledge base is relevant. The doctor observes both subgroups, but to elicit for the population, they need to reweight what they directly observe. So that's kind of the context of this experiment. And we want to examine how well the individuals make these sorts of adjustments. So the setup is similar. but in each day, the number of patients the participants see is fixed, so it's 13, but they're now grouped into 2 subgroups. So we stated that one of the subgroups has a better chance of symptom relief than the other subgroup, but we didn't tell the participants which subgroup. So we've randomized the participants to different splits of patients. So odds of 80 to 20, 70 to 30, or 60 to 40. So the participants were randomizing to each of these groups with seeing different splits between the 2 subgroups, and we've block randomized to the method. So they're still using either the dissection or the chips and bins, but they've been randomized to them. So we've used a high precision scenario, so sampling from a binomial as in the high precision scenario of experiment 1, with probabilities of symptom relief for subgroup 1 and subgroup 2 that are listed here in the slide. So after the individuals, as with experiment 1, individuals were asked to choose amongst the 2 pills of interest, and then once they've chosen, they would observe the outcomes for each of the 2 subgroups. So the pill was equally effective in both of the subgroups. And the target quantities, we're now elicited 2 different quantities. So we've asked participants to elicit for a target population that was representative of the split they saw. So this means no extrapolation is required to elicit this quantity. And then we've asked participants to elicit for a 50-50 split where extrapolation is required in this case. So I'm not showing results for the sake of time for this experiment, just a summary, just because this was less interesting. So there was no evidence that extrapolation or its level affects bias, expressions of uncertainty, or overall accuracy. And participants, there was a slight suggestion, a moderate correlation, that participants that were less accurate in expressing uncertainty without extrapolation were also less accurate with extrapolation. So that's what we'd expect really in these circumstances. So this is just an illustrative slide. So it shows you that the 3 groups have similar spreads, and the mean is fairly close to 0. So we don't really expect, we didn't really interpret these as being any different. So in terms of experiment 3, I think I'm a bit behind with time, so I'm going to try and hurry up. So in terms of experiment 3, so experiment 3 focuses on Delphi. So I don't know if you've heard of Delphi before, but basically it's a method that makes use of rounds of questionnaires. So at every round, individuals are fed back with anonymized information about the group and individual distributions on the previous round, so of other elements of this group. So this is a form of controlled interaction. So you don't rely on the actual physical interaction between people. So you feed back, at every round, you feed back to each individual expert the results of the group to the previous round, and ask if they are willing to adjust their initial elicited estimate and re-elicit again. So the European Food Safety Agency adapted this method, typically used for just point estimates, but the FSA adapted this to allow eliciting for probability distributions. So obviously this method has several advantages. It doesn't require experienced facilitation, and it can be completed remotely. The issue is, despite being widely used, Delphi methods are widely used, it's really unknown how individuals revise their estimate and what they ponder and consider. It's a process that it's not really well understood at all. So we focused on this aspect in these 2 experiments. So obviously with the multiple rounds, the objective is that the final pool distribution is more accurate than the initial distributions that individuals provide. And for that to happen, it's based on the assumption that the less accurate individuals revise more towards the group distribution. So at the end of several rounds, your end distribution is closer to the distribution of the most accurate individuals. So we aimed to test, so we've implemented 2 sub-experiments here that aim to test whether the extent of revision depended on the individual's accuracy. Oh, and also we've tried to understand how individuals revise their judgments in general. So in this case, so participants played a game, so like in experiment 1 with a high precision scenario, and they've elicited exactly what they've observed. So the target quantity was defined as the quantity they've observed. They received 3 types of group summary. So they're randomized to receive one of these scenarios. So if the black is the individual's elicited distribution, then they've received one of the 3 distributions in red. So these were hypothetical, we've defined them. So the first scenario, the first group received a group distribution that's consistent with the participants. The second group received a distribution that is inconsistent. So the mean was defined as the 85th percentile or the 15th percentile of the distribution, but was equally precise as the elicited distribution. And the third group received a group distribution that was inconsistent, had the same kind of deviation than the group 2, but it was much more precise than what the individual gave. So the standard deviation was half of the individual's elicited distribution. So the individuals were then asked, after seeing the group summary, they were then asked whether they wanted to revise their elicited distributions, and if yes, they would re-elicit them. So again, these individuals were randomized to the method of elicitation by section or chips and bins. So briefly, in terms of results, we've had 44%, so 32 out of the 72 revised their initial distributions. And the likelihood, the proportion of people that revised was much higher in the 2 groups that were discordant. There's a slight trend there, but I'm not sure if it's interpretable really. So the likelihood of revision differed by group. The other result, which is not shown here, is that the probability of revision with the chips and bins was much higher than with bisection. So we have 57% versus 32%. So in terms of other results, in terms of the extent of revision, there was a larger changing mean in the discordant group. So this is extent of revision in the ones that revised. So the ones that revised more their mean in the discordant groups. And mainly out of these 3 here, in terms of uncertainty, the participants who saw group summaries concordant with their own, on average, revised to a more certain prior. The ones that saw discordant, but with similar uncertainty to their own distribution, went either way. Some became more certain, other less. And those that saw group priors discordant, but more precise than their own, revised to express a more certain prior, most of them. Critically, there was no evidence that different levels of accuracy to the initial prior affected the likelihood of extent of revision. So the second sub-experiment relating to Delphi looked at, instead of providing the participant with just a single group distribution, so the dotted red line here, it provided the participant with distributions from members of the group, individual members of that group. So these were all hypothetical again. And this experiment really recognizes that the same group distribution can be generated by a very different profile of individual distributions. So in these 2 scenarios, which we've implemented here, you can see that the uncertainty in the group distribution here, the red dotted line, comes mainly from a pool of individuals that elicit with similar uncertainty and similar means. So the uncertainty in the group distribution really comes from within expert uncertainty. You can have a very different scenario where you have exactly the same group distribution, but that's formed by a number of discordant, so the gray distributions here, a number of discordant but more precise individual distributions. So in recognizing here, we've randomized participants to see a number of individual distributions, so they don't see the group distribution, just the individual distributions from other members of a group, and to one of these two scenarios. And we were curious to see how they've reacted to these two scenarios. So the individuals that saw a group that was consistent between its members revised more than those that received summaries from other individuals that were inconsistent among themselves. Again, as with the previous experiment, chips and bins participants revised to a much greater extent than bisection participants. Critically, there was no evidence that accuracy affects probability or extent of revision. So in terms of highlights of these three experiments, participants, as we expected to experiment 1, did not exhibit bias, but do not adjust estimates of uncertainty sufficiently, so they're not accurate in expressing uncertainty. There was a small difference in accuracy favoring chips and bins when precision was higher, and individuals also preferred this method. Experiment 2 generated no evidence that the need for extrapolation affects accuracy. And in what concerns experiment 3, experiment 3 showed that individuals were rational in changing their estimates. So they've typically moved their estimates towards the group distribution. So that reflects that they understood what they were being requested with the task. Importantly, the method of elicitation determines the likelihood of revision, which is an important find. And even more important find is that these experiments found no evidence that the accuracy of the individuals affects their likelihood or extent of revision, which basically casts some questions on the validity and value of the Delphi process. We obviously have to recognize that these experiments are not naturalistic. So they only test knowledge from observation, and they are quite abstract, which was purposeful, but it meant that participants had no wider understanding or emotional attachment to the task they were asked to do. The sample of patients was very homogeneous, which is not typically the case in naturalistic settings. But obviously further research, we wanted to look at a particular aspect of elicitation, and we can think of forms of further research using the setup here to relax some of these assumptions in turn and have a look at what the effects of these were. And I think I'm going to stop here and thank everyone for having the patience to listen to me. And I hope this was relevant and important. And I know it's not directly related to data science, but I think it's very relevant to everyone that deals with data and that makes decisions on data.
Thanks, Marta. That was really interesting. I think it was really relevant to data and data science, actually. So yes. I disagree with you about that. So how do we manage questions?
Yeah, I'm not sure. We've not done this before.
People can drop them in the chat, or they can turn on their microphones.
Shall we see what happens if we just tell people they can turn on their microphones if they want to ask a question? Maybe I'll kick off. So so it seems like people were underconfident in the first, was it in experiment 1? Mm-hmm. When in the high precision scenario, is that right?
Mm-hmm. So across all experiments, actually, because all of them used, so experiments 2 and 3 also used the high precision scenario, individuals were always underconfident.
I mean, do you think in a sort of decision risk process, is that sort of desirable because you want to compensate for the fact that the world is non-stationary and the underlying model that you're trying to estimate might be wrong? So if you're using it for, you know, could that actually be a good approach to be a little bit conservative, or do you think that's always a bad thing?
I think if you assume that in every context you're in a high precision scenario, that could be less serious than being overconfident. So I agree with you that it would be less serious.
There might be protection against sort of the goalposts moving in some real world scenario where you believe you're in a very stationary environment and you're assessing some very stationary target. I wonder if in the real world, the target is often a moving target. And so you want to be kind of robust to like a more online learning sort of scenario or something.
Yeah, so I think ideally what you'd want is that they could be able to make the judgments accurately, and then you as a decision maker will judge whether you want to reflect any additional uncertainties, so make it wider. What this experiment shows is that they are underconfident when there's high precision. And remember, this is relative, so we don't really know what this means. So in the scenario where there was a high precision scenario, they were underconfident, but in the low precision scenario, they were overconfident. So basically what this tells us that people are not really great at judging uncertainty very well, very accurately, and they can't adjust sufficiently when you move the level of uncertainty around. So I guess, again, what you'd like, in direct response to your question, I think what you'd like is for people to be accurate, and then you as a decision maker can inflate that more to reflect other uncertainties that haven't been considered.
Yeah.
I think you're muted, Caroline.
I shall unmute myself. I got loads of questions actually, but I'm going to try to kind of sum them up all into one, maybe two. No, let's try one. So what do you think I think it's the most important one for me. You were mentioning that these are obviously controlled experiments, and that's important because you're trying to sort of understand different sort of mechanisms of reasoning and so on. What do you think the implications are, though, for real life settings? Like, how should we draw from this to sort of influence how we practice?
That's a good question. So I think it depends a little bit. So one of the things, I guess, obviously there's in naturalistic settings, so in you would probably, depending on your context, you'd be probably trying to ask individuals to reason about, for example, a number of sources of uncertainty of knowledge that they might have. So for example, doctors, if you ask a doctor about a probability of symptom resolution in a particular population, will have in mind their own patients, will have in mind their papers they've read, will have in mind conversations with other individuals. So they'll have a number of sources of uncertainty of knowledge that they use. And there might be different biases in kind of having them accessing these different sources of information. But with these experiments, I think by making them so controlled means that basically if individuals can't really adjust their judgments of uncertainty in such a controlled context, I doubt they'll be able to make it in a naturalistic context. So I think the findings here are more about so for example, an experiment 2 where we found no difference between judgments that needed some adjusting and judgment that didn't need some adjusting. So that was a negative find to that experiment. We didn't find evidence of extrapolation affecting the need for extrapolation affecting accuracy. I don't think you can extrapolate those onto a naturalistic setting, but I think the positive finds, meaning finding that they can't adjust their uncertainty representations appropriately can be extrapolated beyond the controlled setting. So for example, the Delphi experiments, the fact that we found no evidence of the accuracy of the individual affecting the likelihood or extent of revision really just tells us that the Delphi process is, I think you can extrapolate that finding there. If it can't in such a controlled setting where everything is straightforward, and if you can't find the benefit of that iterative process, then I'm not sure how you can justify any benefits of this iterative process in a more naturalistic context.
Yeah, okay, that's interesting, thank you.
Any questions from anyone else here? Just unmute your mic if you want to ask. I think anyone who's had reviewer comments from very confident reviewers is aware that the quality of reviewers eliciting information isn't always updated in a way that's proportional to the quality of the reviewer. So there was something there. Okay, I think we'll let people get off to their next Zoom appointment. I'm sure people have days of them, like all of us these days. That was really great for Marta to kick off our virtual seminar series. I want to thank her again, and I think we cancelled a few physical seminars, but maybe we're going to ask the speakers whether they'd be prepared to give virtual seminars now that we know how to do it. So everyone, please keep an eye out for information from the Data Science and AI Institute, and we'll be sending around a newsletter with information, or check the website. And let's thank Marta once more for that presentation.